Hi, we are Molly and Josh, and we're the Modeling Microbial Growth and Resistance team. Thank you so much for coming to our talk as we explain our work with Data Plus over the past summer. In the field of microbiology, the focus of many experiments is to collect growth data from bacterial populations. This growth data is typically calculated from the measure of optical density. The idea behind optical density, or OD, is that light will be shone down onto individual wells of different bacteria trials. The idea is that the more light that can be seen coming through the individual well indicates that less population growth occurred. Once this growth data is collected, it will typically create an S-shaped or sigmoidal curve, as we can kind of see to the right. The results of growth experiments are pretty integral to many types of applications. One such application would be treating infections. The way that growth data experiment might be set up is there might be a particularly harmful strain of bacteria being tested. The different conditions could be different types of medication meant to kill bacteria. The condition that leads to less population growth would be indicated as the more successful treatment in that experiment. A growing field in microbiology is the study of non-standard bacterial growth under stress. This could be studying extremophiles, or basically bacteria that lives in hydrothermal vents under the sea, like high pressure, high temperature, those sort of extreme conditions. Another way of studying non-standard growth would be undergoing experiments that study bacteria with deleterious mutations. That way, that experiment might be set up is conducting one trial with a baseline strain of bacteria and the genetically altered strain. The idea is to see how integral that one mutation is to the successful population growth of bacteria. This type of experiment would be especially integral to the field of genomics, or just trying to map out the entire genome of one particular strain of bacteria. One issue with trying to study these non-standard growth of bacteria is we don't typically have models to accurately depict this type of growth. The standard growth of bacteria will usually follow this like S-shaped curve because this is a result of standard bacterial growth typically following a parametric model. The main parametric model has three parameters: A, or asymptote, lambda, or lag time, and mu, or maximum specific growth rate. As we can see here along the sigmoidal curve, A typically represents the upper limit of the population growth. Mu will typically be the derivative of the inflection point of the curve, or basically the steepest tangent line that could occur along the S-shaped curve. Lag time will typically be the time intercept of that steepest tangent line. We kind of interpret to how much time it takes the bacterial curve to sort of reach that sort of like steep tangent line. Many types of bacterial experiments will include other variables as they set up their experiment. However, most of the time, these many other types of external variables can typically be converted into the terms of these three main parameters: A, lambda, and mu. One way we're trying to hope advance being able to study non-standard growth is with this new model, phenome. Phenome is a hierarchical Gaussian process model. The two main unique attributes of phenome is one, that it is non-parametric. The idea behind a non-parametric model is in comparison to a parametric model, which has a finite amount of parameters, such as the three we see here. In comparison, a non-parametric model will actually have sort of an infinite amount of parameters, sort of like a new parameter for each new data point. And because of this sort of infinite number of parameters, it allows non-parametric models to sort of have a partial amount of ambiguity and allows us to create these more like irregular non-standard curves. For example, we see here below, we have the sort of orange S-shaped curve, which would be indicative of standard growth. However, below it, we have this sort of black, kind of like wavy, bumpy, going up and down curve. This could be a result of a non-parametric model and be indicative of that non-standard growth. The other main unique attribute of phenome is that it is also a mixed effects model. This basically means that it can control for both fixed effects and random effects. The idea behind fixed effects, it is typically indicative of whatever effect of the treatment being incurred onto bacteria. However, with random effects, they're more indicative of sort of a single instance of heterogeneity within a single trial. Random effects might not have anything to do with the treatment being affected, and they could be correlated amongst different wells. Another aspect of random effects is it can occur at several levels. When a bacterial growth experiment is being conducted, it will typically have a single large batch with many different wells of different trials. A single well might include a certain condition, and then a different well have another condition. That single batch might also include many replicates of the same condition being meted out, so several trials of the same condition being applied to bacteria. Random effects can occur between different batches and between the different replicates within a single batch. So with phenome, we can control for the random effects on these two levels, hence why it's a hierarchical model.
So to quickly examine the underlying concepts behind the phenome model, we'll look at two ideas, Gaussian processes and MCMC, or Markov chain Monte Carlo sampling. So to start, Gaussian processes are non-parametric distributions of functions, and they follow a multivariate normal distribution with a mean function, f hat of t, and a covariance function, k of t. This equation under the methodology shows. And then sigma squared variance and length scale and a few other parameters are estimated parameters in this Gaussian process model, and those allow the highest probability distribution of samples to be reached. So MCMC is the sampling process for the models, created using ideas of Gaussian process to estimate the appropriate parameters. So to summarize MCMC, samples from a distribution are taken and new samples are generated based on the previous sample, hence the Markov property. And this is done to get to the highest probability distribution parameters in the samples. So the initial or prior distribution is used to generate samples randomly, and then neighboring parameters are chosen, and then these samples are recursively used to generate the next sampling if those samples with the new parameters are in a higher probability density region. And then the sampling ultimately concludes after either the specified number of iterations or when the highest probability distribution parameters or the distribution converges or asymptotically reaches a stationary average distribution with the highest probability density region and the parameters associated with it. And then this final distribution can be plotted and analyzed with the other statistics. So the code itself that we used uses a special form of MCMC called Hamiltonian MCMC in a Python package called PyStan. So using the ideas of Hamiltonian dynamics and energy conservation, this type of MCMC decreases correlation between the distribution and next samples generated from it. It increases the probability that the next sampling will lie in a higher probability density region. And to do so, there are bigger jumps between the states of the samples as seen in the image between the Hamiltonian Monte Carlo and the Metropolis Monte Carlo sampling. And this is based on the probability densities. So as a result, less samples and iterations are needed to reach that final distribution and parameters as compared to the regular MCMC method. So Hamiltonian MCMC, which PyStan uses, is ultimately more efficient.
Once you're able to understand the methodology needed to run phenome, you were then able to create a workable Python script to actually implement and sample from the model. We were able to set up the code so that the end user would be able to select from various treatment variables from the metadata. They could select many covariates depending on how they wanted to set up their model. With that set up, we were able to use PyStan to implement the actual Bayesian statistics aspect. Using PyStan, we were able to compile a model based on the specifics of phenome. We were able to start sampling from the Markov chain Monte Carlo simulations, and we were able to do some rudimentary diagnostic tests. While implementing their code, we were also able to kind of like streamline the technological procedure needed to actually run the model. Through experimentation, we were able to decide on a appropriate PyStan environment to actually run our code. We decided on Jupyter Notebook. That seemed like the easiest to actually run PyStan. We were also started to like streamline how end users would actually be able to download all the necessary environmental features for phenome. kind of like understanding the easiest way for them to download all the package requirements, allowing them to understand how to actually download all these Python modules, and also kind of like explaining to them the best way to download a C++ compiler, which is sort of a necessary but maybe not always explained aspect of using PyStan.
And after rewriting the code, we compiled our own models, as Molly mentioned. And then with these models, we utilized the Python PyStan package to run MCMC sampling on the models. So the table of values under the results section at the very bottom, those show a few of the model values. This table extended for quite a bit, but those have the values of the three functions, some of the three functions involved in the distribution, and sampled values for the parameters of length scale, sigma, and alpha in the table. And then the plot shown directly above that under the results section, it shows the log result of the 2,000 samples taken within the MCMC that we ran during for this data set. And the mean of the samples can be representative of the model itself to represent the stationary distribution. And then for the next steps of the project, the main focus would be on the deployment of the fully debugged GUI and the code. And then we considered between creating a remote desktop and a web-based app. And specifically, we looked at Jupyter Hub with Kubernetes and other technologies such as Docker to scale these environments. But other options also include the other listed options like Django, Flash, SharePy, and other Python applications to make web apps. Thank you.
So thank you all. This was very, sorry, you done?
Yeah.
Yeah, very interesting. I'm afraid I think we have low attendance as folks are sort of Zoom fatigue. But this is very, very interesting. So do Dr. Schmidt or David have any questions on the technical side?
I just wanted to say you guys have done a great job this summer. You've really engaged with the code and thought deeply about the sort of the biological motivation for this, and that really shows through in your poster here. I'm curious about the deployment options. And I mean, that's one of the things that we've really been struggling with on my end. And you guys have done a lot of work here to look into different options. Which one of these options would you recommend, or would you prefer, and why moving forward?
When I was looking through the Jupyter Hub one, that seemed like it could be a viable option. With we set up a more basic Jupyter notebook on Jupyter Hub. The one problem would be with the pricing with Google Cloud Kubernetes engine. There was I think it was like $100. I forgot the exact number, but something like that to actually maintain all of the user servers to keep them up. But in actually being able to distribute the same version through Jupyter Notebooks itself, it was able to do that. And then also you could use Docker in order to get that actual code on that Jupyter Notebook. And then it was also relatively easy to access because there's options to, like, you could purchase a domain because it's that Jupyter Notebook is hosted at a single IP address. So when the customization was actually done with Docker, it should be fairly accessible. It was just the cost issue, but we were unsure if that same functioning of Google Cloud Kubernetes could be taken on by our own IT department.
Great, thanks. Yeah, and so just so I understand, the Docker container would be sort of set up as a single instance that users would log into and use it. Is that the idea?
Docker was used to initialize the Jupyter Notebook itself. So to access the actual thing, it would be just like accessing a Jupyter Notebook, or you can just use the IP address too. I
see. Okay, cool. And Molly, you were mentioning about or one of you was mentioning in the results about the tables that come out, for example, with alpha and the different parameters. I'm curious about visualization of that. Like, have you tried visualizations that seem to mimic what's in the paper, or was that sort of the next step from here?
For the visualizations of the parameters like the alpha and sigma, we didn't get to those visualizations, but that would be a next step.
Mm-hmm.
Visualization of the sample.
Yeah, cool. And would you envision that also being implemented into the Jupyter notebook?
Yep.
Mm-hmm. Cool. Great. This is really great. Did you have questions for me, or Paul, did you have questions?
I mean, I have the sort of generic questions that I've been asking, which are sort of, do students, getting you all to talk a little bit, especially because this will be recorded for later and it's useful. Just kind of about your journey through the process. So imagine, you're in a job interview and you're sort of describing like how you did what you did, how you got from point A to point B, not on a technical level, but like what was hard, what was easy, what you learned, what was frustrating, like anything you want to share in that front.
I mean, like the first few weeks were mostly just like becoming more familiar with the code, like the basic algorithm. And then I kind of like grew into actually exploring how PyStan works, kind of like using Python in a research setting, being able to use GitHub, learning how to download all the requirements for a new project, just sort of like more familiar ourselves with the process of just like what computational research looks like and how we have to like prepare for all like the aspects that might not necessarily be familiar with us.
Yeah, I just want to raise that just that in your head is extremely valuable. Like it's the lots of data science projects in industry and honestly even in grad school will look, though that scaffolding you just put up there, that's always there. And so having done it once is really helpful.
I'm also curious about process as well. Do you can you two make comments on sort of how you work together and how you who it's a little bit clear to me, but hearing it from you would also be helpful to know sort of who did what and what parts and how you work together on that.
I think at the start, I looked more into looking at the actual code from the MIDS team, kind of like sussing out what the algorithm looked like. And then when we moved on to like working on the Python script, I also like helped debug like some of the issues that weren't allowing it to run.
And more closer to the start, I was working more on the deployment side with looking at the different options. I was able to create a Jupyter Hub thing with Kubernetes and go through that process. And then when we were going through the code itself, rewriting it, Molly and I would work independently and then talk through problems that we had. We conference, have Zoom meetings to try and to get on the same page and that type of thing. And then we were able to get to the visualizations, everything with the code in the past few weeks.
Great. And did you have a certain platform that you used for coding together? So you both had the same version of the code? Did you both write on the Jupyter notebook, for example?
We actually like started with like separate versions of the code. I think Josh like worked in another Jupyter notebook. I think I started in PyCharm, but then I also moved to Notebook. We would typically like use screenshots to like depict like the portion of the code we are currently working on so that we could compare notes.
Mm-hmm. Awesome. It sounds like you two worked really well together.
So it's interesting. You didn't have like a version control set up like Git or something because you weren't worried about sort of overriding what each other were doing.
No.
Okay. Cool.
Yeah, I started them out with like a Git repository at the beginning. I didn't like neglect that, but the thing was they were working on pretty separate stuff, so it was like a lot of overhead for two students.
For two streams.
For one of the doing like completely separate lines of work on the project for most of the program.
Yep.
So yeah, it was a lot to
No, that can be an enormous amount of overhead. Okay, well, thank you all so much for really hard work this summer and just Data Pluses in general.